So I asked you to decide which of the two protons is deprotonated first, which then designates one of two different mechanisms. And so I know you thought about that all weekend long. All right. Okay, so if we go with red, raise your hand, is the first deprotonated proton. How about if we go with blue? All right. I got about 50% participation in that. And of the participation, that's about as much activity as I got for most people. Okay. There was very few. I know. Okay. It is the blue that is deprotonated because in the intermediate that can be stabilized by the ketone that it's alpha 2. All right, and so we are building a pyrimidine, and we now have a base pair that doesn't look like one of the normal ones that we use. Okay, here is where we're going to add the base pair to create a nucleotide. All right, nucleotide has the phosphate, the ribose sugar, and a base pair. And we've got something that is really odd in the intermediate. Okay, we've got a positive charge on the oxygen. How in the world would we draw a mechanistic arrow to get to the intermediate that we see? What is reacting? A lot of times we say deprotonate, right? But there's no proton missing. So what moves? The lone pair. So one of the lone pairs actually forms the double bond. The leaving group is the pyrophosphate, and then the pyrophosphate gets broken down into two PIs. So there's energy derived from that. All right, that is weird to get that particular intermediate. It is especially stabilized by the amino acids within the active site in this particular reaction. And the next, we're going to connect the base pair, but what's between the nitrogen that binds to the carbon of the ring, all right, making that bond, what's in the way is this proton. And so now we have a proton to take, and we will attack, and we will then relieve the strain by putting the electrons back onto the oxygen. All right, so we synthesized a base pair and we added onto the ribose. This is backwards from what we saw with the purine synthesis. We started with the sugar and we added individual atoms on to make the ring. Now, O is not a normal base pair that you would see in either RNA or DNA, and so we've got to get to something that we are familiar with, and U is something that we're familiar with. So we're going to go through a decarboxylation. This is also a very odd decarboxylation, but this is the best proposed mechanism where the electrons will form the double bond of the CO2, and I will pick up a proton. All right. The intermediate that is there, okay, before the attack, okay, where this is the nitrogen, that's right here, and up here is the carbon right there. What you actually have is, in the intermediate step, is a negative carbanion on an sp2 hybridized system. This is an unusual reaction in that there's no stability, but all the mechanistic experimentation feels that this is the best mechanism. There's a couple other proposed ones that would not have that carbanion there, but experimental evidence shows that that is not the case. This is the actual mechanism. And so we have created UMP. That is something that we are familiar with. Okay. At this point in time, we have UMP. Okay, we have TMP and we have EMP. All right. There are specific kinases that will phosphorylate them to the diphosphate stage. Adenylate kinase is different. It can convert it to the di, but it can also then convert it to the triphosphate. If any of them are at the diphosphate stage, they can be worked on by nucleoside diphosphate kinase and phosphorylate them all up to the triphosphates. Normally we talk about triphosphates. So now we have UTP, that is different than the UMP that we saw, and we're going to do one more reaction to get to CTP. Okay, and so we have a reaction in which we've seen this before. Glutamine plus a water, glutamate, and the amine coming off to become the nucleophile. So we've seen that several times. And it is powered by ATP. I won't show this mechanism. We've seen a couple mechanisms very similar to this. And we have CTP. So at this moment in time, we have all of our RNAs. ATP, GTP, UTP, CTP. All done, except for one more base pair. Okay? And the base pair that we have to synthesize is what? What's the T? Okay. Now, here we go. When we have all of the RNAs, if we want to get to DNA, we've got to do one thing. And you can see this really simple reaction. Take that hydroxide, and it's going to be a hydrogen, right? And unfortunately, this is not the mechanism, but it looks really easy that I could say this. A hydrogen, or hydride, comes in and attacks at the top. That falls off as a proton. Okay? That doesn't happen. Why? Why would you say that doesn't happen in this case? Do you see the coloring? If this hydride attacked at the top and this fell off, this hydride sits up on the top, right? Well, look at this. Black, black. Here's the new one. It's on the bottom. So it can't just be an attack like that. And so this is actually a very complicated reaction, even though the overall reaction looks simple. Here's the enzyme. It's tetrameric, but it has two subunits, two R1s and two R2 subunits. And there's a lot going on in here. All right. There are a disulfide pair. That's important. Okay. We also have a, what is this amino acid here? Tyrosine. But what is that tyrosine seen as? A tyrosine what? Radical. Okay, so there's a radical reaction. Okay, so there's also a couple sites. The active site where the reaction happens is right here, between the two interfaces. Then we have a regulatory site, okay, and then we have a specificity site. Because the specificity of the substrates is ADP, CDP, UDP, and GDP. That is unusual for enzymes. Enzymes typically will bind one thing, very specifically. That's one of the things that we talk about enzymes. They're very specific and bind one thing. What happens is, depending what is bound in this regulatory specificity site, one of them bound will then be connected to one that is bound inside the active site. So if I have, let's say this one, I'm going to have one of these, and if I bind here, I'll have one of those. If I bind here, I'll have another one. And so they are controlled by what's there. All right. This is usually the primary regulation site is the availability of energy, and ATP is one of those drivers. All right, let's take a look at what happens in there. All right. Any base pair can be bound between the R1 and the R2 subunit. And the first thing that happens is this X radical reacts with the hydrogen of the 3 position. Wait a minute. What in the world? This 3' hydroxide and that doesn't change at all. We're supposed to be working on the 2'. So that's weird. We're reacting with the 3' position. It's the hydrogen of the 3' position. Remember, this radical was a tyrosine. All right, and 1 electron will pick up the proton, and 1 electron will stay as a radical on the carbon. Okay, that makes this whole ring structure a little bit more unstable. Next, the water will deprotonate the sulfur. What you can't see really well, and I'm going to articulate, is there is a negative charge now on the sulfur. And we have the protonated water, which is a pretty good leaving group, which they don't show, but the bond will leave, creating a carbocation that is next to a radical. That is a very unstable reaction. All right, here's where it gets tricky. This sulfur that was deprotonated to negative makes a disulfide bond. What leaves is a hydride group. The hydride negative reacts with this positively charged carbanion to create the deoxy form. And one of the last reactions with the actual ring is to react back with the tyrosine and this radical to terminate the radical on the ring and creating and regenerating the radical on the tyrosine. And now I have the deoxy form. But the enzyme is functionally dead at this moment in time. Why? Why would this series of events, if I had this flowed off and I had something flowed in, why could it not do another reaction? What is it? The radical is there and it can react, but what else can't happen? The disulfide bond. Not just the fact that we formed the disulfide bond, but a hydride left, right? And I need a hydride to create the deoxy form, and so that is missing. Once this floats away and before this comes back in, I have to reduce this back because it's oxidized. Okay? This is where the NADPH comes in. And so the dead enzyme has to be regenerated into a live enzyme by taking this disulfide bond and reducing it. Now we said NADPH does that. Well, it does by giving it to an FAD, which then gives it to thioredoxin, which then gives it to the ribonucleotide in one case, or in another, I will give my NADPH, break a bond of a, what is GSSG? Do you remember? Or GSH? Glutathione, good. What is glutathione, though? Do you remember what it looked like? We taught it. It was a little tiny peptide, but it was an odd-shaped peptide because it had 2 C termini. And it was there were 3 peptides. One was put in through the R group as the peptide bond, and we had a sulfur group, and it's really high in concentration inside the cell so that it can donate hydrides. And so I will give my hydride to glutathione. Glutathione will go to glutaredoxin, and then it can reduce. Okay. Normally, we go through thioredoxin, and that's the normal pathway, although this is a very important reaction. Very rarely do we have a backup, and here's the backup. When another pathway was found when a mutant didn't contain the thioredoxin, glutaredoxin was found to be the carrier. And so there's an alternative pathway for this to create the dioxys. All right. Now, interestingly enough, the regulation, I don't want you to know what turns on what, but to know that certain things that are binding in the specificity pocket will change the affinity and the specificity, and there's a balance that is kept. And so don't memorize what forms what, just know that you have to have different components to create different or all of the deoxy components. All right, one interesting thing. Tell me about this structure here. What is odd about that structure? Okay, lean over to your neighbor, ask them, what in the world, why is he asking me about this? It's a nucleotide. What in the world is wrong with that nucleotide? Or is there anything wrong with that nucleotide? Is there anything odd about it? Anybody see? That particular nucleotide. Okay. Name for me the base pairs in RNA. A. And what else? U and G and C, right? Name me DNA. A, G, C, and T. Okay. Is that RNA or is that DNA? That is DNA. DNA U. Does that exist in our genome? No. Okay. But it is an intermediate for the synthesis of T. Okay. So it is synthesized temporarily and then converted into the deoxy T version. Okay. And so UDP is converted into DUDP. You take off a phosphate, and now we can do the thiamine synthesis. All right, what in the world is different between this? Okay. That's the same, that's the same, this is the same. Oh, a methyl group. Okay. Methyl group has to be here, and there's not one there. That's the only difference between U and T. Okay. So how do we do that? Tetrahydrofolate is a what carrier? A methyl carrier. Okay, that seems to be pretty good. Now, this is carrying it in a different way. We saw a methyl carrier by putting a methyl group on here, just a methyl sitting here. This is a methylene. Okay, so that's different. We also saw tetrahydrofolate carry a formal group over here when we did some synthesis. And so we added an aldehyde to things as one carbon carrier. But what we're going to do is we're going to move this methylene here and make it a methyl group, methylene to methyl. Ooh, that seems like magic. Okay, and so let's take a look at this. All right, once this is generated, we'll take a look and talk about that the dihydrofolate that is produced has to be regenerated to tetrahydrofolate, and then we have to put another carbon onto it. And so there is a regeneration step. All right. Here is where we introduce the first concept and one of the first chemotherapeutic drugs. Okay? Because what is a cancer in just very general terms? A rapidly growing cell, right? And so if you are rapidly dividing, you have to rapidly reproduce everything, including what? DNA. So, in this case, at least this is generalizable to all cancers. All of them have to produce DNA. If you can block the production of one single nucleotide in the DNA synthesis, and there's none of it around, or very little of it around, the reproduction is going to be halted. Okay? This looked like a wonderful drug that could cure everything, and we found that it didn't, but it is a good start at a chemotherapeutic entity. It's a little generalized. Okay? And so we're going to stop rapid growth by targeting the synthesis of DTTP. Okay? And we're going to do that at both the enzymes, the one that synthesizes it and the one that regenerates the tetrahydrofolate. Okay, I'm going to show you the two pathways. All right, the normal substrate is DUMP, the deoxy UMP. Fluorouracil is the chemotherapeutic drug. Okay, what is the difference between the natural substrate and the drug? Do you see? That one fluorine. Fluorine is not too much bigger than a hydrogen. It sits right there. But we're going to have a whole lot of consequence in that particular position. All right, so it binds into the enzyme. It is in the active state, in the active site. The tetrahydrofolate is also in the active site, and the enzyme can start to do its job. One of the active site amino acids is a sulfur. They will both attack at the carbon of this double bond, and we will create a carbanion on the carbon that will have the methyl. Okay, and so at this stage, they're exactly the same. In the next reaction, that negative charge can be a nucleophile. It will attack, well, there's also something that happened also. Inside the enzyme, the tetrahydrofolate had a lone pair form a double bond and broke the bond between the methyl, the carbon, and the nitrogen. So it's sitting in this Schiff's base form. The negative charge will attack. The negative charge will attack. The electrons will kick up. The electrons will kick up. Okay, no difference at this moment in time. Looks exactly the same. All right. And so here, the next step after that bond has been formed, after this bond has been formed, I use a base in the enzyme to deprotonate, right? I take off the proton. I form a double bond. I have a base. What happens? There's no proton to deprotonate. This is an inhibitor. What kind of inhibitor can we classify this inhibitor as? Well, first off, is it reversible or non-reversible? Non-reversible, okay. And have you heard the terminology suicide inhibitor? Where it becomes covalently linked in the enzyme and it's totally ineffective and dead. That enzyme cannot be regenerated. The only thing the cell can do is to break down that protein, go back to the DNA, and create a new one, okay, which takes time. And if we're rapidly growing, that's not something that we can hold on to. All right, let's finish what the reaction occurs in the normal. After I have deprotonated and formed the double bond here, then I can have the electrons in the tetrahydrofolate form a double bond. A hydride can attack at this double bond. Electrons can get back down to form the double bond, and then the enzyme sulfur leaves as a leaving group, and now I have thymine. All right, and so if I stop the reaction, I won't make any of that. Additionally, this dihydrofolate not only donated a carbon, it also donated a hydride, so I have to reduce it. Give that hydride back. This enzyme, dihydrofolate reductase, well, when I give fluorouracil, we also give these drugs. Why? These drugs will bind into the active site of this enzyme and stop that enzyme from regenerating. Based upon your knowledge of the structure of tetrahydrofolate and these drugs, what kind of inhibitor do you think these are? They are competitive. They actually have very similar shapes to the normal substrate, so it's just going to get in and bind in the active site and slow down that enzyme. All right, and so a lot of times when we use chemotherapy, we use not a single drug, but we use cocktails. Lots of drugs at one time. Okay? Now, here's the bad thing about these generalized chemotherapeutic drugs. They attack every single cell. They will stop growth of every single cell. Okay? And that has consequences. Well, most of the times, most of your cells aren't dividing very rapidly. Okay? The ones that are dividing very rapidly, these affect more often, okay, and have a greater effect against them. But we are giving it to the entire body. There are some cell types in your body that do grow, controlled and regulated at the rate of a cancer. Can you think of any of those places inside your body that your cells are dividing very quickly? The lining of your stomach. Okay? And so when you take chemotherapy, you can become nauseous because you're affecting those tissue types that are in your digestive tract. Where else? Hair follicles. And so a lot of generalized chemotherapy, you tend to lose your hair because you're attacking just fast-growing cells. You're killing all cells with these drugs. It's just most of your cells can tolerate it, some of your cells cannot. And we reduce the tumors, hopefully, and normally we reduce the tumors so that they can be operable and so that we can take them out. Newer anti-cancer strategies tend to do this, and we want to do this. We would rather target exclusively the tissue that has the tumor growing and not do generalized all over the body. And so a lot of different very good drugs have been developed for certain types of cancer. Not all cancers have specific treatments though. Okay. Now, another reason why we do chemotherapeutic cocktails is this. We tend to challenge the body with a very high concentration of drugs very quickly, very kind of really hard, because we want to attack them aggressively. But what happens is there is a generalized transporter in every cell that tends to take anything hydrophobic in the cell and pump it out. Well, that's normal. Sometimes we get stuff inside our cells and we want to pump it out. Okay, but they're targeted mainly to hydrophobic molecules. Now, if by chance you go to the doctor and you get a strain of resistant antibiotics and it's resistant to penicillin, what does the doctor usually do? If your infection doesn't respond to penicillin, you get a different class of antibiotics. A whole different shape, a whole different mechanism by which the drug will attack the bacteria, okay? Well, in chemotherapy, we're treating you with these very high aggressive drugs with very high concentrations, and what happens is this protein that pumps organic molecules out of the cell sees that challenge and then starts to proliferate. Makes lots and lots of copies of the what's called MDR transporter, the multiple drug resistance transporter. And so what happens is you give a whole bunch of drug in the bloodstream. They get absorbed inside the cell. You get the concentration up to a therapeutic concentration in the cell so that you can stop TMP synthesis. But if your cells start overproducing the transporter, then what happens is you get more and more and more transporter. It will pump out anything that's not supposed to be there, that's organic. And so you could give the patient a ton of drugs in their bloodstream, but the concentration inside the cell doesn't raise very much because as soon as it gets in, it gets pumped back out. You become immune to the cocktail. Well, the answer was, well, let's try a different drug. Well, if you get this kind of resistance, you don't get resistant to just one drug, you get resistance to all drugs, okay, or mainly hydrophobic drugs, which most of our drugs are hydrophobic. And so we tend to hit very hard with lots of drugs at one time for short periods of time. And so you have treatments or courses over long periods of time to try to stop that resistance from forming. Okay. Now that we've made them all, we can talk about degradation. We'll talk about purine degradation, particularly because there's a disease associated with it. Nucleotides are always undergoing constant turnover, and some enzymes that break things down. A nucleotidase will remove all the phosphates to get to nucleosides. That's a base pair and the sugar. Then the nucleoside phosphorylase will use a phosphate coming in, and it will displace the nucleotide, the base pair, away from the sugar, and the sugar will be phosphorylated. So the ribose comes off as ribose 1-phosphate. And then you've seen the enzyme before, phosphoribomutase, that converts ribose 1-phosphate into ribose 5-phosphate. Where did you see phosphoribomutase before? What pathway? You might have seen it in the pentose phosphate pathway. Okay? And so once you've got it into the ribose, you can then take it all the way through the pentose phosphate pathway and you can degrade it. Okay? But the nucleotides have to be degraded particularly. Nucleotides have lots of nitrogens. Nitrogens, if given off as ammonia, are poisonous, so we have to have a pathway to degrade these. All right, once the two nucleotides are cleaved, you got GMP up there, you got AMP. You can change AMP into IMP, and you've seen that before. And then we cleave the phosphate off to get to the base pair and the nucleotide and the sugar. And we can get GMP phosphate to get the guanosine. Use the phosphorylase to break off the sugar and the ribose 1-phosphate leaves. We now have two base pairs that are floating around, guanine and hypoxanthine. The conversion back down to xanthine is a commonality if I take off the amine from guanine, and if I oxidize hypoxanthine, I get xanthine, and if I oxidize that one more time, I get a molecule called uric acid. Uric acid is the degradative pathway by which we get rid of the purines, and they are filtered out through the urine. It's got a pKa of about 5.4. Okay? Here is the bad thing about this molecule. It has a very low solubility. So you can't have a lot concentrated in the bloodstream. Okay? If not, it starts to form a salt. Okay? This salt is not so good for us. And so, in the degradation pathway, we use a suicide inhibitor. Okay? Allopurinol is the drug. It's done by xanthine oxidase, which do these two reactions. And it will, normally here, it'll put this carbon into a ketone. It'll do part of the reaction, put the hydroxide, but not finish it up. And once this is formed, it binds to the enzyme so tightly that the constant of dissociation is almost the same as if it was covalently linked. It's not covalently linked, but it binds very, very tightly to the point that it doesn't dissociate. The dissociation constant, which sometimes are seconds, is like in days. It will let go of it over one every few days. And so it stops that enzyme from being produced. Okay? Why in the world would we ever do that? All right? This is the kind of salts that are created. They are needles. And they tend to form or collect inside the joints. All right? In the fluid and in those cushioning places between your joints. What if you had your joints cushioning little things and then you just put needles there? How do you think your patient's going to feel? They ain't going to feel very good. Okay? Now, this disease called gout is typified by a particular joint that it's seen most often in. I have no idea why, but people find the first joint pain happen in the big toe. Yeah, why the big toe? I don't know, but that joint is very susceptible, and it's very typical of gout. But the pain is so much that they can't even walk, many of the patients. It's that bad, okay? And so what we tend to do is, these salt crystals also will affect joints and then the kidneys. Kidneys are very susceptible because if the salts are going through in those very tiny capillaries, it can really tear up that tissue type, and so that's not good. Why people do have gout, believed to be genetic, but that biochemical cause has not really been well elucidated. We just know that we have patients have this.
So I've heard that eating a lot of red meat
Yep.
can cause gout.
Yes.
Why is that?
When you eat the red meat, you also you're eating all the cells that bring in all of that DNA that also has to be broken down. Again, too, when you bring in and eat a lot of meat, if you brought in sugar, you can store that, right? If you bring in fat, you can store that, but every amount of meat that you bring in has to be processed, has to be broken down. There's not a storage for the protein itself. Yes? You give the drug and you reduce protein consumption. And so the drug will then stop it at this point, that molecule, and this molecule, okay, of which can still be filtered out. They will not form the salts. They're more soluble than the final product. These tend to have issues with kidney because you're filtering out something that is not normally filtered out. But if we reduce meat consumption, that tends to also help. Why in the world? Yeah.
Is there a way to reverse what's already there?
Once you get there, no. Once it gets to that reaction and that it's a pretty stable, salt.
So
There's not a good way to reverse it. There's not an enzyme that drives the reaction back the other direction.
once you have it, you just have it.
Yep, and you have to filter it out. And what it is once you start filtering it out, then the concentrations go down, the crystals will dissolve. And so you can get it out that way. Why in the world do we even have this particular molecule? Urate itself is really a very strong antioxidant, much better than even vitamin C. And so there are benefits for having urate in your system, and it doesn't necessarily, it is not having that antioxidant versus it isn't immediately toxic allowed this particular molecule to be formed.